All right, so welcome everyone to the 5th lecture and seminar in computer architecture. I'm Mohammed Alser. I've been with you in the first lecture. For those who don't know me, I'm going to introduce myself briefly in the next lecture. For today, we'll talk about totally different topic. We call it genome analysis. And we will show you a bit of the efforts we spend in our research group at ETH on how to accelerate genome analysis, either using algorithms or hardware accelerators, or a co-design of both of these. Normally, to get the best speed up, you need to design an efficient algorithm that is aware about the hardware that you are going to use, and then you design the accelerators based on what you have invested in the algorithm development. Okay, so I'm working as a lecturer and senior researcher at ETH since September 2018. I got my PhD from Turkey, Belkent University. I received the IEEE Doctoral Dissertation Award. And then during my time at ETH, I'm fully focusing on bioinformatics application, try to mix it with the new algorithms, developing hardware accelerators and so on. So if you're interested in continuing in this direction, please feel free to contact me, Onur, or anyone in the research group. We have a large number of PhD students, master's students working with us on this very specific topic. We have Joel here, we have John Fertina, Konstantinos also may be interested in this topic. And we have very good experience in the past courses, like project and seminars, seminar in computer architecture, for example. We got a lot of students after finishing the course. They were presenting some papers about bioinformatics topic, and then they show some interest that I want to continue working on this. What should I do? Then we assign a project. They keep working on them for papers or for their own curiosity. Okay, let's start. So what is a genome? Look at this picture, what you can see. What are the interesting things that you can observe once you just look at this at the first glance? Excellent. So there are things that you can observe just looking from the shape, from outside, right? You don't know what is inside the body, but there are things that you can observe. Hair color, eye color, body shape, mass, all this stuff, right? And there are interesting things that makes us different. We are really fundamentally different, right? It's not about white and black. It's not about being from Europe or Africa, but it's about things that for our own things. So we own these things that makes us different. So what are these things that makes us different? Is a genome. What is a genome? So it's a complete set of genetic instructions. So think about it as a recipe book where you want to cook something. You go to that recipe, you fetch it, how to prepare a cake, for example. You bring the ingredients, then you activate some of these ingredients, either to include or exclude based on your flavor or taste. You want more sugar, more salt, so you activate more sugar, more salt, and so on. So exactly same thing in genome. We have a lot of genes, a lot of things as instructions. But whether they are activated or not, that is a different question. But all of us have a gene. Either we pass them to our kids, and they might have these phenotypes, and that's why I prefer to call them phenotypes, not a disease, because, for example, eye color, it's not a disease, right? Just different color, but it's not harmful. And exactly the same thing in the genome. We have a huge number of characters, and collectively, all of these makes a genome. A lot of genes, if any gene is enabled or activated at some certain age or at some point in your life, or even is not activated but pass to your kids, then they might get all of these, the things that we can observe just by looking to the outside of this human being. If we look closer to the genome, if we print it, if we convert into computer science problem, as you need just input text, for example. What does this mean? What does all of these A, C, G, T? So the first thing, as all of you smart people, you can observe is we have only 4 characters, A, C, G, T. What are these? So these are representation for the chemical molecules that make our DNA. What are they? Adenine, guanine, cytosine, and thymine. All of these are chemical molecules bind together to make the chain of our DNA bases. Now, If there are some differences, for example, this C over there, you can use this, the C over there, or A, or even a large chunk of your DNA is different between individuals, then you can infer, okay, these what makes this disease, these what makes the eye color, because these are different, and I know from the phenotype, I can observe that this eye color is different from this eye color and so on. So when I compare the DNA together, I can observe some of these causes. Now, who knows this? All of you, right? What is it? Prime tower, excellent. Yeah, so this prime tower, how large is this prime tower compared to our genome, our DNA? Who said smaller? Who said larger? Think about the DNA. We want to print it on A4 page, very small margin. We want to print the font size 10, spacing very small. I want to stack all these papers together. Which one smaller, which one larger? Prime tower is larger. Smaller, okay. Excellent. Close. So prime tower is about 120 meters, but the stack of all these pile of papers is about 140 meters. So almost there. So our DNA collectively together, printed in all this paper, it's really larger than this tower. And this, you can think about the huge workload that you need to process during using today's computer. How about the other species? This very small virus, the DNA is about only a few thousand characters. This tiny virus can infect this bacteria and cause harm to this bacteria. And this bacteria DNA is about few million characters. Where are we? We have few billion characters, 3.2 billion characters in our DNA. How about this? There's a red onion, has 5x longer DNA than ours. How about this? This Japanese flower has about 50x longer DNA than ours. So you can see the diversity. It's not a small workload that you can play with. You can design simple algorithm to do efficient analysis. You really need to spend time to do careful analysis very efficiently with an efficient algorithm with very low complexity. Of course, the fruits, vegetables are well known for very large genomes. They have really complex genome. Okay, now we show the benefits or the importance of reading our DNA so that we can know the causes for all these diseases. How to read the DNA, basically. Ultimately, we want to have a sample of our DNA, want to give it somewhere, and that black box or whatever will give us this full sequence of our DNA. However, we don't have such thing. There is no magic that can take the sample, give you the full sequence of your DNA. What we do have, go to pharmacy, I want to analyze my DNA, give me a kit. They will sell you this kit, $100, maybe $200. There's some promotion these days. You can get it for more or less. But regardless, you can order it online and get it. What you can do, you take this small tube over there, swab it, swab whatever, blood sample or from your nose, and then post it back to the company. Same as you do COVID testing, but rather doing it at home, you need to send it back to the company. Why is that? The company will have one of these very expensive machines. These really wide diversity, wide differences between all these machines. This is fridge size. This is a huge machine. This is bench top. This is handheld. This is a small gadget for the iPhone. So it's really a different thing. However, all of them share one same property, regardless this machine, regardless the price, the technology you use. What is it? They give you pieces of your DNA. They just generate randomly subsampled from your DNA pieces, either short or very long. And the bad thing, unfortunately, about these pieces is that all of them lack any information about the order and the location. We don't know which piece comes before which piece. We don't know where's the location of that piece in your DNA, so that we can say, okay, let's link them here, here, here in this order. We don't have such thing. So current sequencing machine provides small randomized fragments of your DNA. And this is the main problem that we are going to explain in today's lecture. All right. Yes, please. That's a very good question. So to answer that, we really need to show the steps for sequencing. So before you do sequencing, the sequencing is actually these machines. We call them a sequencing machine or a sequencer. Before you send the sample to this machine, there is a step called library preparation. So you need to prepare the sample, make it understandable for this machine to read. During this library preparation, what you would do is to fragment the DNA. You will cut it yourself. So using some ultrasonic signals or just rotate in very fast speed, and then you will shred it out. You will isolate the plasma, some content of your blood, and then you will have only the cells with the DNA. Then you shred the DNA so that you will be able to read it using this sequencing machine. This is one of the main causes for this randomness. You don't have any control once you cut these pieces. Now, you yourself also will cut it into different lengths. So you are the cause for the problem, or the guy who's working in the wet lab while he's doing the sample preparation. Why he cut it? So during sequencing, they add some enzymes for the sample so that it do chemical reaction with the DNA fragment itself. For example, we know A always should bind with T. C always bind with G in your DNA. That's how you have the chain of DNA. So these chemical molecules, they have hydrogen bonds in the last orbit, and to get chemically stable, they need to bind together. So A always bind with T, the C always with G, and vice versa, G with C and T with A, right? So one method, one technology to do the sequencing is to add A and wait for chemical reaction. Whenever you observe a chemical reaction, you know there is a T at that location. Same thing, you add C and wait for chemical reaction. Once there's a chemical reaction and you added C, then you know that there was a G at that location in the DNA. I'm not going to explain this, but if you're interested, I can show you some slides from other lectures in project and seminar course. I can show you in detail how it's done. But this is how is the process. And to add these chemical molecules, they have a lifetime. So you can keep repeating adding these chemical molecules to the DNA, but after some time, these chemical molecules get weaker and then they cannot react. That is the lifetime of the enzyme added to the reaction. And if you know that the lifetime is going to take this few milliseconds, then even if you have longer DNA, it won't work. Just give you the first few pieces of the DNA, and then you will lose the rest. So in order to exploit this DNA, to try to read as much as possible, we divide it into very small fragments first, then we give it to the machine will read the full segment, and then we do some computational steps that are easier than chemical reaction to try to infer whether this piece come before that piece and so on. Does that answer your question? Okay, perfect. Okay, now we agree that all these magic machine or sequencer or chopper, whatever you want to call them, that they give you randomized pieces of your DNA. Now, what are the properties of these data coming from the machine? Now, we know that all of them ACGT data, just text file that you can read it using your own computer. Now, the reads, every piece coming from the sequencing machine, we call it a read or a read sequence, okay? The reads can be three types: short, very long, and long but accurate. The short pieces are 100 long and very accurate. 99.9%, I'm very confident about every single base I give you in the output. And only about 0.1% of the time, the sequencing machine is not confident about what it provides to you. We have also on the other side, very long reads can really vary from 100 all the way to 2 million or 2.3 million character. In the short reads, all of them are 100 long, all of them same length. In the long, they're really wide diversity. Could be from 100 all the way to 2 million long. But there is a serious problem with the long reads. There's at least 15% inaccuracy in the output. So 15% of the time, I'm not sure if this is A or C or G or T, even if the sequencing machine telling me this is C, but it could be just a noise, right? There's a third type called hi-fi reads, high fidelity read. This type of reads, the length of the pieces can be just between 10,000 all the way to 30,000 long, cannot reach millions. However, the accuracy is very accurate, 99.9%. Yes, please. Sorry? So these sequencing technologies. So this, for example, machines coming from Illumina. Illumina is one of the sequencing companies, use certain sequencing technology, and always the output is this. This is from Nanopore. This is from PacBio. Okay, now PacBio has another type of read that is similar to long reads. We call it CLR. Okay, this is CCS or hi-fi reads. This is the latest technology in sequencing. It can mix the advantages of this being long and the advantages of this being accurate. However, the price is still expensive. This is very expensive. This is very expensive to have. And I have here real flow cells. Seems we have technical problem. While we fix it, maybe you can have a look into this. You can take one. Take one. This is what we call a flow cell. So that is used for the sequencing itself. You need to inject some chemical molecules. You take a sample with this. You take a sample from the chemical molecules. You add it to that piece. This piece you have in hand is very expensive. It costs $900. It wants one-time use. You just throw it away once you do the sequencing. That's why the sequencing is very expensive. To read your DNA, you need to pay at least $2,000 using certain technology. For other technologies, it might be a bit cheaper. For example, Illumina, you can pay a few $100s. You can see this piece, only this piece is $900. That's what we call flow cell. If you don't believe me, you can go to Nanopore website, go to reagents or flow cells, go to R9.4, check the price. Okay, this is what we call flow cell. Again, it's one-time use. There are also chemical molecules you add, or what we call reagents, that you'll need to pay for it as well. And then you add the chemical molecules over there, and then you need the sequencing machine. This is not the sequencing machine. This is just flow cell. You need to have the sequencing machine, and the sequencing machine, you can keep it. It stays forever, as long as it's not broken, okay? So that's what we call a sequencing. Maybe people on Zoom, I don't know who can show them this piece. Maybe you can hold it on next to the camera. Yeah, I don't know if they can see it. This is the advantage of being in the classroom. Yeah, but at least you can see it online. I can send you the picture later on over Moodle. Okay, and there is also a sensor in the back, if you observe it, similar to the camera. You can see it. Or this is part of the hardware connected to the hardware we have in the sequencing machine. All right, so what we do with these pieces, since they are really randomly sampled from our DNA, they are useless. We don't know if this is coming from chromosome 1 or chromosome 2 at this location, so that we can say, okay, the eye color gene is located in this chromosome, so we need to find whether you have A or C in that location. We have no such luxury information to know. So what we do, exactly how we solve the puzzle. We need to have a picture, or what we call a reference picture, and we need to have all these pieces coming from the sequencing machine. And then the problem is, or as a computer science problem, we want just to compare every piece and guess if it is coming from here, comparing with the reference genome, and said if it is high similarity, probably it's located here. And we keep doing the same thing with all of these until we link all pieces together with probability. Of course, we are going to make some mistakes. All right, so as a curious computer science student or electrical engineering, I wanted to do something with the data. Give me the data. Okay, I'll tell you to go to this link, download this file. It's about 1 gig compressed data. Once you decompress it, you'll get about 3.2 gigabyte of data. That's what we call reference genome. Once you open it, you will see something similar to this. That is the ID. It could be the sample or your name, whatever, you can control this. And that is the actual content of your DNA. This is, you'll get about 3.2 billion character in the file, separated by chromosome. So this is chromosome 1, you'll get another ID for chromosome 2, and then the content, chromosome 3, the content, and so on. Why these are N? We didn't say anything about N, we say ACGT. N is when the sequencing machine cannot read this region. It will give you something undefined, which is N here. That is the reference genome. What is the reference genome? Is thing to use as a reference when you collect these pieces or when you link them together. So this file is the next thing I showed you already that we call reference genome, and it's representative for each species. So if you move to viruses, you need to have a reference genome for that virus. If you move to bacteria, you need to have that reference genome for bacteria. How to get it? Go again to the same database. This is from NCBI, NIH in US. Go to assembly, and then you have a search. You just insert bacteria, virus over there. You get list of assembly or reference genomes for all these species. Yes, please. Very good question. So we pay a lot to get just this file. We pay about $3 billion, 13 years, 13 years of research just to get this file. That is back in 2002, 2003. We got the first draft of what we call a human reference genome. That was really a huge achievement. A collection of consortium efforts from Japan, US, UK. I don't know if Switzerland was included in that time, but it was a huge effort between many countries, and trilaterally cost us $3 billion. So every character read in your reference genome cost $1. Because we have 3 billion character. And until now, we cannot get the complete file. So as I said, these are gaps. These we cannot read them. Until now, we cannot get the complete thing. There was only, during the last year, one paper called A Complete Human Reference Genome. You can search for that paper. I think I have one slide about it. And that solved all chromosomes except chromosome Y. We're still missing chromosome Y. We still have some gaps there. So think about, it's not 13 years of research, but also 13 plus 20. So about 30 years of research just to get near complete reference genome. Yes, please. That's a very good question. So normally they do assembly between multiple individuals. It's not single human being. It's multiple individuals. They try to increase the confidence by having multiple samples, and then they collect this information with too much overlap so that they can build the confidence over linking these pieces. So for example, as I said, the sequencing machine, as any machine, it's not perfect, right? They introduce their own errors. So again, even if this is A in your DNA, the sequencing machine can tell you this is G. So to resolve these errors, sometimes they overlap the pieces together if they have the very high similarity together, but very few differences. So they overlap them, and they said, okay, once we overlap, we do majority voting. How many A in that location vertically? If the majority A, then we said, okay, probably the rest are sequencing error, and let's have it as A. So they collect these from multiple individuals, not single individual, okay? And that is a very good question, because also as a human being, we are curious, what if I compare my pieces to your DNA, right? Who tell you the piece I have at this location is similar to your DNA, such that I can link it, right? That's a very interesting question. And that's why we have a field of research that we call pangenomics. Pangenomics is that I want to build multiple reference genomes for every individual in the population. But again, it's a very expensive process. It's not easy. So we want to build this reference genome for every individual, or at least a representative of populations. So I know population or people living in Switzerland, they share somehow the same lifestyle, or maybe share some characteristics or share some ancestries. So let's say, at least at fine grain scale, it might be good to have one reference genome for people living in Zurich, or people originated from Europe, or people originated from Africa, and so on. That is where we go as fine grain scale for the analysis or for the comparison. Yeah, this is the database I was mentioning, SRA from NIH. Here, the search space, if you type whatever you like, you'll get this information, and this is the data you can get. For this exact link, this is the read set. The previous link was reference genome. This is the read set, the things you got from the sequencing machine. And you can get information about what type of machine was used, for how long it was running, how many bases you got, and all this information. What is the project was about, and the sample number, the run number, and all this information. And here where you can download the read set, you will get what we call a FASTQ file. In the reference genome, we got a FASTA file. I don't know if I have it somewhere. Either FNA or FASTA file, that is the format of the file. For the read set, we always get a FASTQ file, okay? Now, you can download this data set, publicly available. Once you get it, you need a tool that is, again, computational tool, just a program, to do read mapping. Read mapping is the process where we link these pieces together, where we compare the read set with the reference genome, and then we said, okay, this piece coming from here, this piece coming from there. How we do it? We want to map the reads to a known reference genome with some minor differences allowed. Why we allow for these differences? Because fundamentally, my pieces are different from the reference genome. So we want to allow for these differences. Plus, there are sequencing errors. I want to tolerate these. This is the DNA molecule in molecule format, chemical format. I give it to library preparation and then sequencing, and they got these pieces. And then I have the reference genome. I compare every piece with the reference genome, and then I decide on these pieces. What is the right location for every piece? Of course, I can get multiple locations for every piece. And then the challenge, how to decide what is the right location for that piece. Either you can follow some metrics like, I want the location that has the least number of differences with the read I have. And then if you still get few, you can randomly pick, or you go more advanced level and you said, I want to do dynamic programming such that I know what type of differences I have, and then I can prefer certain type of differences over others. What are the possible type of differences I can have? Can have substitutions, insertions, or deletions. These are the three type of differences can happen in my DNA. There are some other large or different types of what we call large variation or large differences, such as inversions. So the DNA itself could be inverted. It could be a large segment of it deleted, large segment of it inserted, as we will see later on. Okay, yes, please. That's a very good question. I think I have one slide towards the end to summarize this talk. But yeah, very perfect question. What if we have a sequencing machine, you give it a sample, treat the full DNA? What will happen to all these algorithms, hardware stuff? Any answer? Very valid question, right? What will happen to all these? If you check fundamentally what's happening, you are comparing a small piece to very large piece, right? And to try to infer where this small piece is located, right? Now, if I get, instead of small piece, very large piece, and I want to compare this large piece to another large piece as genome to genome, how I should do it? Somehow similar to this method. Of course, I need indexing first, that is hash table, so that instead of comparing as a brute force with every single location in the reference genome, I can compare only with the small portions that share high similarity, for example, with the large genome. And then I will reuse some of these components, some of these algorithms, some of these methods, right? Not as is, maybe, but with some minor modifications, right? Yes, please. That's an excellent question. Why we need to do that if we read the full DNA? How you are going to infer the causes for disease? How you are going to infer what you are sequencing? Think about, I'm swapping this table, I will send it to machine. I want to see what is on this table, right? The machine doesn't know what I'm sequencing. It might give you the full genome or collection of genomes because there are many things on the table, but doesn't tell you what kind of virus they have. So I need to compare to many reference genomes as genome to genome so that I can say, okay, with high confidence, I know that this genome is similar to this genome, so probably you have a virus, you have this bacteria, you have this species, and so on. Okay? Even individual to individual, human to human. I want to see the differences between you and me, or me and all the people in this classroom. All right, so I downloaded the FASTA file, downloaded the FASTQ file. This is the read set again. This is the reference genome. The reference genome, one ID, and then the full content of that chromosome or that strain. In the FASTQ, it's 4 lines per read, 4 lines per read. This is the ID. This is the read itself, only this line. And this is, again, can consider it as ID or whatever. Normally just plus sign. We don't have this because it's repeated of this, but you can consider it as this. And this is the base quality, the quality of the sequencing machine while generating this. So this is an ASCII code. Each character here is an ASCII format. If you convert to ASCII, you can get the percentage of the confidence when generating each character here. So each character should have a character in the base quality line, okay? These are useful information. Sometimes we can leverage them and we delete the beginning or the trailing, depends on the quality. If the quality is very high or very low, then we can ignore a portion of this because it's useless, right? The sequencing machine might confuse you, tell you this is A, but it's not. Anyway, this is the content of the read and this is the reference genome. What we want to do, we want to compare them together. So how we do that? We extract pieces from the reference genome and we store them somewhere in the index, in the hash table. Why is that? Because I cannot search a huge book by reading every single character and compare. Is this similar, similar, next page, similar, similar? No, I cannot do that. This is a very expensive operation. What I do normally, I have an index in the beginning of the book. It will tell you the chapters or the section, which page, right? And if you're interested in certain portion of that book, you go to the index, retrieve the page number, go to that section, and so on, right? Same thing here. If I want to search very large text, the first way to do indexing. So you need to index that text. So you take randomly or certain method or following a sophisticated algorithm in a way how to pick these pieces. And then you store the piece itself or a hash value of that piece. And then you store the location of that piece. So the hash table will be like 2 columns, data structure. First column is these pieces. The second column, the location of every piece, right? Now the time to get the read, I get the read, I extract pieces from the read, same way as I'm doing in the reference genome. And then I query every piece in that data structure. Give me what are the location for that blue piece. I got this and this. Give me the location for the green guy, this and this. The same thing, the location with the red piece, and I got this and this. Now, what makes sense to me, I said, okay, in this line, these pieces are followed by each other, right? So the blue come before the green comes before the red, and so on, right? So I would assume these pieces to appear exactly the same order in the reference genome so that they are similar to each other. So give me where are the location where I have the blue followed by green followed by red, which is here, right? I can see the same order is maintained in that piece. And then probably I said, okay, with high confidence, I have this piece similar to this, then this might be located in that location. I go to the reference genome, I fetch that line from the reference genome, and I have it here. And this is the read. And then these pieces, the things I was fetching from the previous slide. And then what is remaining is just to check the similarity between these pieces, the gaps between the seeds. How many differences, how many similarities? And that's what we call base by base comparison or sequence alignment. What is sequence alignment? One method to do sequence alignment is to use edit distance metric. There are too many metrics to be used here, as Hamming distance, Needleman-Wunsch, Smith-Waterman, for example, and so on. There is a lot of formulation on sequence alignment, how to compare sequence with sequence. For edit distance, if someone wants to define it as the minimum number of differences that makes one sequence similar to the other. So I want to do all modification to that sequence so that I make it similar to the next, and then I count the minimum number of these operations needed to make both of them similar. Let's take example, the best way to explain anything. I want to make organization similar to operation. What should I do? Yes, please. Yeah, exactly. Can you explain it? What's going on here? Excellent. Excellent. And these are the operation that I'm doing. And once I count this operation, that is the edit distance, if it is the minimum. If it is not the minimum, this is not the edit distance. For example, I can delete the 2 characters, insert this chunk of characters, maybe I have some substitutions and so on. Now, the edit distance for organization and operation is 7, based on this example. Take this another example, organization and translation. So I have 3 ways to do it. Which one is the edit distance? Which is? The third one. Perfect. Excellent. So the third one provides you the minimum number of edit operation needed to make this word similar to this word. That's why the edit distance is 4. Apparently, in this case, we have multiple ways to do the edit distance calculation. And then which one to pick? There is no smallest in this case, right? Both of them 7. It's 7, but which one? Which way to do? Yes. Excellent, excellent. I hope people in Zoom can hear the answers or questions. So what she said is that instead of dealing with all these edit operations as similar cost, we need to penalize them differently, such that we can favor one over the others, right? For example, here, there's a huge gap. And here, there are 2 gaps. Which one to prefer? A huge gap or small gaps? Here, there is deletion 1 and another 4 deletions, but here, 5 consecutive deletions. Which one to prefer? That's the game based on the user or based on the penalty score or penalty metric. Yes, please. Exactly. So it depends on what's your purpose of this study. What kind of application? Are you studying protein? Are you studying DNA? Are you studying disease that cause large deletions, insertions, and so on? But this is a user configurable parameter. So you develop the tool, you let the user, whatever the user, might be expert, might be just out of curiosity, right? You provide this capability to him or to her to change all these parameters, and then that is a perfect tool where you have flexibility in all these parameters. That's why sometimes we use edit distance, sometimes we use Hamming distance. Hamming distance is just a way to compare the substitutions, how many substitutions I have. They cannot detect, for example, insertion deletions, which is very important for certain disease. Okay, until now, assume we finish indexing, hash table, some filtering based on the order of the seeds, and then do some sequence alignment. That is a complete ReadMapper, for example. How about the performance of the ReadMapper? So if we check the sequencing machine, the things that you just saw with the flow cell and then the sequencing machine with this very expensive guy, they can do very well. Why? You can see from the cost. They are very specialized to do the job in hand. They know nothing except just sequencing the DNA, right? And for that, they are very specialized to do the job. They can do 48 human genome in about 2 days. That is single human genome in 1 hour to read it fully. Of course, it doesn't sound as easy, but I'm trying to simplify the things. All right, to do the analysis, including read mapping, it's a very expensive job because for every read, you check a lot of locations and then you check the similarity. The similarity calculation itself, it's very expensive to do edit distance calculation, for example. And for that, you need to suffer a lot to do the analysis. You can do only 1 human genome in about 32 CPU hours. So to finish this workload, you will be way lagged behind. You cannot cope with this workload. What is the need for speed? I should care. I can leave the machine 2 days. I'm not that hurried to get my genome in about 1 hour, right? Why we need it very fast? Think about this application. So here we have a bunch of individuals. For each individual, we have a piece of their DNA. So that is 16 piece over there. And we have one of the disease that we are interested in as a blood pressure. Might be disease, might be something else like eye color, not necessarily disease. Again, I want to correlate these pieces with the health information. What are the codes for blood pressure or high blood pressure? Again, all of you smart people, you check, you have everything here similar, but there are a few differences. Yes, please. Excellent, perfect. So based on SNP1, that is single nucleotide polymorphism, single difference in your DNA. If you observe only that information, you'll directly correlate it. If we have a T, that is directly correlated with low blood pressure. But whenever we have a C, we have high blood pressure. If you look at the second guy over there, total random. Whenever we have a G, it could be low, it could be high, right? So that is what we call personalized medicine. So knowing your DNA can prescribe a medication for you based on the things you have in your DNA. Not all medication suitable for everyone. If you think that disease can be caused by single variation, that is wrong. Look at these diseases: autism, obesity, underweight. They are caused by a deletion of a huge chunk of your DNA, 500,000 bases from chromosome 16. Same thing with obesity, and even you can see the opposite disease. Underweight and obesity are caused by the same amount of DNA altered, either as a deletion or a duplication. If you don't believe me, here are the references. This is a paper, you can read it, published in Nature. There's another paper, paper, and paper. Yes, please. I don't know if there is a reason why this very specific number, if this is your question. Honestly, I don't know. I don't know. Yeah, I cannot answer that. Yeah, I don't know. It could be with high confidence that this right amount correlates to the disease. It could be something else. Could be mixed with sequencing errors, maybe more or less, but at least they observed this region with the people they were studying. Especially for autism and cancer, these are very complex disease. There's no single location in your DNA cause these disease. So what we call, I think I have another slide explaining what we call GWAS, genome-wide association studies. Yes, please. Very good question. Hopefully I have that slide. No, I don't have it. Yeah, I have it in another lecture. How to do this study? Normally we do genome-wide association studies. What does it mean? We have two groups of people, one as control, one as cases. One healthy of people, they don't have that disease, but they might have something else, right? Same as your question. How do you know that this location is correlated with this out of very long genome, right? And they have another group that is not healthy, that they have the disease, and another group healthy, right? So now I get the DNA of all these two groups of people, and then I compare the differences between them. And then I plot what I call Manhattan plot. A Manhattan plot is an XY. The x-axis is the chromosomes or all location of your DNA, and the y-axis is the frequency of every difference. How many times I get A in that location in your chromosome. That's why I call it Manhattan plot, because it looks like Manhattan Towers, New York. Now, based on the frequency, I can observe above a threshold, if any difference is happening above a threshold, that means that difference is significant. Now I can get many differences as significant, right? Because I cannot isolate people from other disease. I cannot get perfect group of people that they have only that disease and everything else is similar. So based on that, I do just some statistics. I said, okay, based on my previous knowledge, I already assigned this variation to that disease. So I'm going to exclude that variation from the new disease I'm studying, okay? So you can increase the confidence with that variation by increasing the number of people included in the study. That's why the more people you include, the better. That's why we have a lot of governmental initiative to have a full database of everyone DNA living in the country. So you don't need to repeat these studies. You already have the DNA ready in the database, fetch all of them, study all of them at once. Then that is the best or ideal scenario how to study these variations. Okay, hope that answered your question. All right, so now we agree that this problem is really complex. Can be single variation, can be very large variation in your DNA. This is another example on the importance of studying the genome. This is a real example. If you go to this link, this is a publicly available database called OpenSnap, single nucleotide polymorphism, and this is only for single variations, okay? If you open this link, you will see this window first. What does it tell? If you go to chromosome 19, to this location in the chromosome, you will get this thing over here. What does it say? In your chromosomes, it's pairs, right? One of it coming from the mother, one of it coming from the father, okay? If you have at chromosome 19, a T from the mother and a T from the father, then 20%, if you got hepatitis C, you may get better after getting medication. Only 20%, very low probability to get better. But if you have a C and C, then 80%, you may get better. That's what we call, again, personalized medicine. So before I prescribe any medication to you, I check your DNA based on some historical medical records. I would say, okay, you might get better in 1 week if you take this medication. You may not get better because of this medication, let's change it to something else. Rather than let you suffer, try this medication first for a few weeks. And then once you come back, I said, okay, this doesn't work for you, let's try another one, and so on. That will save a lot of time, a lot of inpatient time and cost. One serious application, when we talk about critically ill elephants, these elephants normally, they have few hours to few days to survive. If they didn't get the right medication, they will pass away. And that's why in some hospitals, they do what we call whole genome sequencing. And if you go to a hospital, they'll give you two options. You want to do it quickly and pay a lot, or you want to do normally and you can afford the price. And the difference in time is really huge, 2 days to 5 days. If you have a lot of money, you would pay and get the quicker option, right? But if you don't have, you will survive. You will suffer, sorry. Doing the whole genome sequencing for critically ill elephants, what's wrong with the critically ill elephants? Why not the doctor see the symptoms and then directly say, okay, you have this disease, get this medication? In those sick people or sick children, the symptoms are not clear. They are mixed up with everything. They can get fever just for any reason. It's not an indication of any disease. That's why it's very difficult to know the disease from the symptoms. And the only way to do it accurately is to read their DNA. Once they do the whole genome sequencing and they get the medication, that will avoid the morbidity, will reduce the hospital stay length and the inpatient cost. And again, if you don't believe me, here are the references. You have few papers to check. Yes. So there are steps in doing whole genome sequencing that are performed in the wet lab. These are, let's say, require your hand job and so on. So you need to really produce this library, bring it to another machine, shred it, fragment the DNA, wait some time, add chemical molecules to process it, wait a few hours, you cannot reduce that time, and then move it to the sequencing machine, and then you'll get the pieces. Once you get the pieces, then the computational time starts, and that's what you can accelerate. But probably if you change the library preparation protocols, you might get another protocols that can do the job in 1 hour, okay? Or in a few hours. But at least as a computer scientist or as an electrical engineer, what you can do is the hardware side or the computational side of it, right? And someone else as a chemistry or as a physics, as any people working with the enzymes as scientists, they can develop something efficient at that side. So that's why in bioinformatics, it's really interdisciplinary work. It's not based on us only, but there are also people needed from biology, from chemistry, from physics, who need to develop their own efficient method for the sequencing, for example. But inside the sequencing machine, there are also hardware. For example, how you observe the chemical reaction inside the machine, you need CMOS sensors. And then some electrical engineers need to provide a very fast CMOS sensor to observe the light emitted from the chemical reaction, or the heat emitted from the chemical reaction, or whatever the technique that you are going to develop as a new technique. Now, because of all these benefits we got from whole genome sequencing, in the UK in 2019, they decide any critically ill infant admitted to hospital, do directly whole genome sequencing for them for free, starting from 2019. And that is something ideal to have everywhere in the world. Now we do also some studies for studying the microbiome. Again, giving the cities, I want to understand what kind of bacteria I have in Hauptbahnhof, in very crowded places, in New York downtown, for example. In somewhere where I have a lot of people, and if anything harmful, then I'm going to harm a lot of people, right? That's something interesting to study as a public health. This is a recent study happening in 2021, where a lot of cities participated, including researchers from ETH and Zurich. They studied all kind of microbiome as viruses, bacteria that are unique only to these cities, just to make signatures for these cities. And they include 60 cities from all around the world, around 5,000 samples, and they find something unique to Zurich. If you're interested in this, they have a very nice web interface where you can see the map, list of viruses, list of bacteria, and where they are located. Okay, hopefully by now we got enough information to motivate you why you should work in this area, why you should care about bioinformatics. You have the expertise to develop algorithms, you have the expertise to develop GPUs, FPGAs, hardware accelerators, or processing in memory. And it's the right time to jump in and invest all these skills in such an important field. Okay, what makes the analysis a bottleneck? Why it's slow? Why it's very slow? The sequencing machine, we agree, it's very quick because specialized, but how about the analysis? As the sequencing machine is developed more and more, and the cost of it is reduced significantly, instead of this having it for $900, if it's become $100, then everyone will start just sequencing for fun, especially if you have enough money to do research about it. And for that, we are going to have a huge amount of data every year because the sequencing cost drop very fast, but the Moore's law is still somehow stable or is not scaling as fast as in sequencing machine. Again, we have specialized machine that they don't do anything except only sequencing. but we are still using general purpose computers to do the analysis. That's very inefficient. We are still using the von Neumann model until now. Until now, we are separating the memory or the data where it's stored from the computation. So we need to move the data from here to here to here, and even we need to move it from the sequencing machine all the way to the storage. And that's very efficient. It will take about 60% of the entire energy spent in the system just in data moving, not in computation. And that's why data analysis is performed far away from the data, which is very inefficient. And here's the paper mentioning that 60% of the energy just spent on data movement. We didn't do computation yet. Once we go deeper into the tool used for read mapping, what is happening inside? We did this analysis ourselves, and we find that about 60% of the total execution time is spent in comparing the two sequences, the very last step of read mapping, when we do base by base comparison, which is dynamic programming step. And based on this theoretical analysis, we cannot do better. You cannot do better than quadratic time algorithm for comparing the data or for calculating the edit distance. Another observation we made that 98% the time we compare the two sequences together after fetching it from the reference genome, and now we do base by base comparison. 98% of the time they are highly dissimilar. They are not interesting to us. So just flush them away. Why is that? Because we use only small pieces in the read to do the comparison with the hash table, right? So we don't do it at the scale of the read. That's why there might be a lot of differences between every two pieces. Now, some of you could say, what if I increase the number of seeds extracted from the read? That's a valid point to do, but it will increase the execution time, because every time you access the memory to fetch this seed and the location, you are spending a lot of time and energy going to the main memory, fetching this information and coming back. Because again, these random accesses to the hash table, you cannot guarantee that to have some certain locality with the hash table. Okay, so if we look at the compute stack, I think you already saw this figure before in the seminar course, or maybe computer architecture. So this is the computer stack. And here at the hardware level or at the data itself, you can do a lot of great things, starting even from the data itself without playing with the algorithm or the hardware. Just if you compress the data, for example, there is a lot of repeated content, a lot of A's next to each other, then a lot of C's, a lot of G's, right? If you do just simple run length encoding, might reduce the amount of data, and then you can do the comparison without decompression, for example. This is one way to improve the things without touching the algorithm. If you move further, for example, this one example from somewhere else, matrix multiplication. Now, I can solve this matrix multiplication using Python, and it will take this time. But I could also solve it using AVX or C implementation and see the huge difference in execution time just by changing the programming language. The algorithm is exactly the same. Everything is exactly the same. The data is the same. The problem size is the same. Just the programming language plus some support from the hardware using some SIMD cores. So that tells you the huge benefits of working at the full stack, all the way from the data to the problem algorithm to the hardware itself. Another motivation, if you are not there yet, reading the genomic data itself alone, just reading the file, fetch it from the SSD, see if using C programming language to read that file, it will take this much of time. That's if it's compressed. If it is not compressed, it will take this time. Compare it to Python and see there are at least 4x speed up just because I'm using C to read the file instead of Python. Think about other steps. That's just reading the file, not doing any computation. There's a link here, by the way. You can play with the code. You can see the differences yourself. Hopefully, with all of this, I spent more than 1 hour just motivate you that we need intelligent algorithm and intelligent hardware to do all this very efficiently. Hopefully, I did my job. Now, looking at the algorithm itself, or what kind of steps we have in genome analysis, we have really a huge number of steps to be performed just to do the analysis after sequencing. So you start from reference genome, you get the reads. There is one step I didn't tell you about called base calling. For example, these technologies, this very expensive guy, don't give you ACGT. They give you electrical current. They give you amplitude, varied based on the ACGT. If it is A, maybe the amplitude is this. If it is C, then the amplitude is that. And then we use very expensive algorithm based on machine learning to do the base calling, to convert these signals into ACGT data. So again, you can think about other computational steps that happening even before do the analysis, just to convert this information to ACGT. And we have a lot of research from our research group only in this step, only in base calling. Once you get the read set, the reference genome, you start what we do, the read mapping. So you do the indexing. This is one-time job per reference genome. So you don't touch it after you build the hash table, done, right? And then you get the read. Sometimes you do read correction to correct these reads, as the machine will introduce some errors to this read. Then you do the mapping. Then you do the variant calling, for example, if you're interested in detecting the differences that cause the disease. Or you can do taxonomy profiling if you're interested in knowing what kind of species you have in the sample, what kind of virus or bacteria you have. That's all collectively we call a genome analysis. And we have this paper from our group going through every detail on read mapping and list all possible algorithms that can be used for doing read mapping. So if you're interested in knowing more about all this algorithmic development, what kind of directions followed there, what kind of things you can improve, I would recommend this paper to read it. I have a lot of pointers, so up to you if you want to read it, or just keep it until the end of the semester, then they can join us for research, and then we can discuss all these papers together. This is the paper again, same paper. All right. So in summary, what are the contributions that we are doing in our research group, Safari research group? We're trying to build accelerators plus efficient algorithms for multiple steps of the things or the pipeline I was showing you in the previous slide. And we did things for CPUs, we did things for the main memory, and we did also things for the storage itself. So there are multiple works as hardware accelerators, just GPU and FPGA that to replace the CPU, where you can move the data from the main memory to these accelerators. We have also some work on processing in memory or near memory. And we have also near memory for doing sequence alignment, but also this year we have something near storage or inside storage that I'm going to show you in a few slides. So the first work called Sneaky Snake. And what is Sneaky Snake about? So we observed that sequence alignment is very expensive, which is where we do base by base comparison, sequence to sequence comparison. And our goal is to accelerate this step by reducing the need for dynamic programming algorithms. As I said, based on that theoretical paper, we cannot do something better than quadratic time, right? You agree on this, based on that paper. Now, what we can do, instead of using this dynamic programming expensive algorithm all the time, what we can do, use something else that is quicker and tell you if these are similar or not. I don't care about the minimum edit distance value, for example, at this stage. I care only about roughly how many differences are there. And then I can tolerate some margin of error. And then I said, okay, I don't need the dynamic programming just to tell me if these are similar or not. Once I find them similar using this magic algorithm, then I can use dynamic programming. And by doing so, I reduce the number of times I need dynamic programming algorithm. I did not substitute it. It's still there. I just reduce how many times I call it to examine the similarity for me. So I use something heuristic, very fast, does not give me the same job as the dynamic programming, but at least can tell me if these are similar or not. Let's see more details. So we observed that genomic strings, once you compare them together, could be similar or dissimilar, right? They might have very high similarity or very high dissimilarity. If we get these guys, that is unfortunate. So we need to ignore them because the number of differences exceeds certain threshold. And based on the disease I'm studying, I said, okay, these differences might not represent that disease because I know the disease is caused by single variation, for example, why I should worry about 4 variation or 5 variation. Just ignore it. If I get similar strings, I will keep them. And here where I apply the dynamic programming. Ideally, if I have that magic algorithm to do the filtering for me, I need to query and then send it to that algorithm and then do the sequence alignment or the dynamic programming if I find it good. And for that, I need the filter to filter out most of incorrect mappings or most of dissimilar sequences. Why most? Not all of them. Otherwise, it will be perfect algorithm, right? Which is dynamic programming. I cannot do a perfect job by using something else. It must be dynamic programming algorithm. That is by theory, right? That's why I'm aiming to filter most of them, not all of them. Of course, I will have false positive, but I cannot tolerate to have false negative or false reject. I cannot reject similar sequences, and my algorithm can say these are not similar. This is not tolerable. You cannot allow for that. That's one of the requirements to have. And I need to do it quickly because this is the goal. I want to reduce the time spent on sequence alignment. How we do it in practice? Yes. Excellent. So at this stage, when I compare base by base, I don't care about all other locations. The problem is only I have 2 sequences, compare them for me and give me the edit distance, for example, right? That is a mapping, one mapping. So whatever I used before, it tells me that read might map to that location. It might map to other location as well, but at least I pick one by one. I pick that location and try to do base by base comparison. If good, keep it. If not, move on to the next location and so on. All right. So this is a full picture of a huge DP matrix, a huge dynamic programming table. This is after I build it. This is one sequence, this is another sequence, and every dot here has a meaning that is similar. So the character here is similar to the character there. That's why I have a dot over there, right? Also, the chunks over here could be similar, or the chunks at this direction could be similar. Once we get something like this, we can say like, there was inversion in the DNA, for example. That's why you have this in the opposite direction over this. Yes, please. Yes, yes. The largest so far, because the largest read we can get is 2.3 million, right? You can build million by billion, which is still huge, right? Very huge. But in today, main memory can accommodate that. And you don't need to build it all by once, right? Because I think I'll explain it. Or at every cell, to calculate this specific cell, you need this cell, this, and this. This is what you need to build any cell, just 3 cells preceding to that. So either you build it row-wise, so you build one row, and then you build the next, and build the third, save it somewhere to the storage maybe, or main memory, and then move on to the next row, right? This is what actually you need. Okay, but look at the full picture, and I have a reason why I show you the full picture, not zoom in snapshot of the table. What is the reason why I show you this picture? I want to observe if the 2 sequences at this axis are similar, then you will always get this kind of diagonal matches, right? You may get some random matches scattered around the diagonal, but at least you will get most of the matches going around the diagonal, right? And this is very, very important observation for me. Correct alignment is a sequence of non-overlapping long matches that are the things around the diagonal. How I'm going to use it? The key idea is to approximate the edit distance by having it or by solving it using single net routing problem. If any of you study VLSI design, okay, perfect. Yeah, in VLSI design, we have a single net routing problem. What is it? You have this VLSI chip. If you dissect through it, you'll see something like this. What are these? So you have the silicon around, you have some pins, these for connecting input and output. And you have some modules on the chip, right? Or system on chip, where you have a huge amount of modules over there. And single NAT routing problem, you have an input there, you want to connect it here with the least cost. What is the cost? Could be latency, could be the minimum wire length, for example, to connect these modules together. Or it could be I want to pass from here to there with the minimum distance. I don't want to go through too many routes over there. And this is how I use it for solving the edit distance problem very efficiently. How I do that? I build a table or a matrix using this equation. Don't worry about the equation. It's already reported in the paper if you're interested in it. But think about there's a way to build this table. And I get this. And now I have an E value. That is the edit distance threshold. So this is the maximum amount of edits I'm allowing for. I want to study a disease that caused, for example, by 3 edits at most. So going from this matrix, which I built it using the equation I mentioned, I convert into this. So every 1 convert into black box, every 0 convert into white box. Okay. What are the boxes? These. The white one is where you can pass. The black box where you cannot pass. It's just an obstacle. You need to avoid it somehow. Okay. Now the problem, or a single net routing problem, I want to pass from here, going to the other side with the least number of movement. I want to keep going in the straight line or with the least number of obstacles. Okay, that is the constraint here. Not the cost, not anything, but just with the least number of obstacles. And keep in mind that every obstacle you avoid is an edit operation. So the maximum number of edits or obstacle you avoid, that is your edit distance, or something around the edit distance, because again, this is not a perfect algorithm, but something that does not give you false negative. It always can decide correctly on similar sequences. But for dissimilar sequences, we don't know. It might reject, might accept. That is still fine. All right, let's solve this problem. Remember, you have 3 trials to try with. You have a snake. I want to solve this problem efficiently. It go row-wise, row by row, and count how many white boxes you can pass through. 0, 0, 0, and here 4 steps. And here single step, 0 step, single step, right? As a greedy choice, which path to follow? The one can send you further with the least amount of obstacles, right? That's how you can get the minimum number of obstacles to avoid. So this snake will choose greedily this path. And how many obstacles I have? 1 here. So I'm going to reduce this by 1. Now it becomes 2. You have 2 trials now. Repeat the process. Count how many obstacles you can avoid. 0, 0, 4, 0, 2, 0, 0. I'm going to choose this path because it can send me further, the furthest I can move, with the least number of obstacles. That is 4. Now reduce this into 1 and move on. Repeat this process until either this is 0 or I'm reaching the end. That is the algorithm. Very simple. Now I reach the end and it's 0. Then it means the algorithm, the sequences used to build this matrix are similar because they have number of edits less than the threshold. Now, how this is faster than dynamic programming? I still have cells as in dynamic programming table, and I still do some computation. Look at this picture. How's this different from this? While I'm building this, once I face an obstacle here, there is no need to build this entire row, right? Because there's obstacle, I cannot move any way further. So just once I recognize this, move on to the next row. And this is what I do here. Once I face an obstacle, move here, obstacle, move, here, no obstacle, keep going until you face obstacle. Then you move here. And this is what you actually build for the sneaky snake. This is the actual table you need to build, right? And you can think about the amount of work you save by not building the entire thing. The other thing, there is no dependencies here at all. I told you, whenever you want to build a single cell in the table, you need to check 3 cells, right? And based on these 3 values, you build this. Here, there is nothing such that. Everything independent of each other. You just compare character by character when you build the matrix. When you build the matrix here, you compare certain character with one of the sequences that is at index I and index J. If they are similar, then it's 0. Whenever it's not similar, it's 1. Whenever this character equal to this character equal to that, this character equal to this, all zeros. And for that, you are comparing only individual character. Give me the first character from the first sequence with the first character from the second sequence. They are similar, put somewhere 0 here. Yes, please. Yes, yeah, we have the proof in the paper for optimality and runtime and correctness. That is a must for us. And that's why we said the requirement for any filter, 2 things. First, ignore or detect most of the dissimilar sequences. The other thing, which is more important than the first, to not ignore any of the similar sequences. And without doing so, then your algorithm might ignore some of the correct sequences, right? And the third thing is, of course, the quickness. You have to do it very quick. That's why we build hardware accelerators for this algorithm. Yes, please. Yes, that's a good point. So we don't know this in advance. You need to build the matrix first. And while you are building, you know that this is the further you can go. And then you don't know all of this. All of this is still not yet to be built. You didn't build anything after checkpoint 2. You only build these, right? You keep building row-wise until you face an obstacle, keep going until you finish all rows, right? The number of rows here, by the way, is 2e plus 1. E is this value, 3. So these should always be 7 rows, okay? So you know the number of rows in advance. Now you reach checkpoint, but you don't know it if it is checkpoint or not. Once you count how many white boxes you can move forward, that will tell you the checkpoint. What is the largest path has white boxes? That is your distance to the checkpoint. For example, here it's 4 plus the obstacle, then that is your checkpoint. And then you iterate from here. Starting from here, you build everything yet this to be built. You didn't build this. You don't know it in advance. You need to build all these rows, 7 rows. Then you find this path and here. You might get another question. Yes, please. Might be it. Excellent. These are some of the ways to improve this algorithm, but we don't do that. What we do is just we stop once we get an obstacle, right? Once we got the obstacle, just stop calculating that row. But for example, we stop here, right? We stop calculating this row, but we have to restart here because we don't know after the break or checkpoint what's going on. For example, here, there are ties, right? How to break the ties. There is here one distance, here one step as well, right? Which one to pick? All of them same distance. All of them single white box in the path. Which one to pick? Doesn't matter. You can pick the one you like, right? For example, maybe I reach the exit in the first row. Doesn't make sense to build the rest, right? There's one way to improve the algorithm. You don't need to do that, but we don't do that for a simple reason that we are doing FPGA. In FPGA, you need to hard code everything, right? You cannot do special cases for certain paths. Otherwise, you cannot replicate the instances doing the same job, right? So we always, for example, each instance can calculate 16 steps forward And then you replicate it many times, as many as you have in the rows, and that will give you everything in parallel, in one shot. That's one of the reasons. But in GPU, for example, you can leverage such optimization, assuming that you have multiple threads. Each thread can go through rows, and each row can build independently these things. But at the end, you need to synchronize. So you'll have the same challenge, maybe, right? Yes. Again, we have to agree, all of us, that all these algorithms should not provide any false negative. That is false reject. That is two similar sequences, and you said these are not similar. That is not allowed. You cannot tolerate that, right? So we have to agree on this. Now, in the other side, what are the accuracy, or how to tell if this algorithm is better than existing algorithm or not? By looking at the false positive. How many times you are telling me these two sequences are similar, but they are not? They are different, and you are producing them to the dynamic programming. If you are calling the dynamic programming to calculate the similarity between the two sequences, that is bad, because you are increasing the time you are doing dynamic programming, right? But if it is a few, as we have here, we still have false positive here. We have false accept, okay? That is not good, but we have it very minimal. Less than 1% of the pairs accepted falsely by this algorithm. And we have the quantification exactly in numbers in the paper. These are another algorithms developed by our research group. And this is when we implemented a single module, only single module, without replication in FPGA. You can think about how low resources it requires. Very, very low number of lookup table needed from the FPGA side to solve one problem. That's why you are allowed to replicate these modules many times to do many problems in parallel, because it just requires less than 1%, right? As a result, we got significant results as speed up compared to other methods. I'm not going through it. Now, how we can alleviate data movement. This is still FPGA and GPU. We have both implementation, GPU and FPGA. We have also SIMD, and we have also processing near memory for SneakSnake. All of them on the same GitHub repo, if you're interested. In this work, we try to improve further SneakSnake by doing the design using a near memory acceleration. We still FPGA acceleration, but we use HBM, where we have high bandwidth passed to the FPGA. The memory itself and the FPGA are coupled together within the same package, which makes the data movement less, as in DDR4, for example. And the problem is that read mapping is heavily bottlenecked by data movement from the main memory. And the solution, we need to perform this quickly and near where the data resides. So we redesigned the accelerator logic of SneakSnake so that we can leverage these recent FPGAs. This is the Power 9 system, where we have the HBM FPGA. This is HBM plus the FPGA coupled together. And this is where we have traditional FPGAs with DDR4. And when we compare the results, you will see significant improvement in both runtime and energy efficiencies. I think you see this figure in the previous lecture, right? So that is for different number of PEs, or processing elements. So this is the replication, how many times you can fit, or how many times you can solve, or how many problems you can solve in parallel in one time. Here, 4 problems, 8 problems, 12 problems, and so on. And you can see the scale, how the HBM can scale very well compared to DDR4, because you have limited number of channels to feed in the data. You cannot feed more data than you have in the FPGA, for example. And then if you're interested, I would recommend you reading the paper. And this is a Grimm filter, another filter doing exactly same job as SneakSnake, but different algorithm, different technique that is using 3D stacked memory. So in 3D stacked memory, we have in each layer a DRAM layer. And each DRAM layer, we have banks. In each bank, we have this thing where you have rows and columns where you can read row-wise through the row buffer. You activate that row, you fetch the data, then deactivate, and so on, right? So optimally, if you want to read with high bandwidth, what you would do, you would read a row from this layer, a row from that layer, a row from this layer, all going to the logic layer, and then you do processing at the logic layer for all these data in parallel, right? So now the challenge, how to first do the data layout, how to distribute the data among these layers so that you do the filtering for all these sequences in parallel. This was the challenge for 3D stacked memory. I'm going through the algorithm quickly. So assume this is one of the sequences or the reference genome itself. Doesn't matter if you pick short sequence or very long sequence. You will pick a portion of the data and consider it as a bin. And out of this bin, we build a bit vector. What does this bit vector represent? This bit vector, as it says, bits, right? Only 0 or 1. Whenever you have 1, it means this portion of data exists, as you can see here, A, so I record 1. Now 1 is overlapping piece, so I start from the second character all the way to C. And if it exists, I record 1. Now what are these? These are all possible values out of a subsequence of length 5. So how many of them? For each option, you have 4 options, either A or C or G or T, right? And the length is 5. So 4 to the power 5 or 4 times 4 times 4 times 4 and so on, right? That is the length of the bit vector you can have. And this is sorted. So every bit here has very specific meaning. So 1 in the beginning, it means this very specific chunk exists in your DNA. And now what you store in the 3D stacked memory is only this bit vector. Right? You don't need the data itself, the DNA data. And you can think about how much you save by just changing the representation of the data, right? You move from ACGT content into this bit vector. And now you can do the comparison between the bit vector very quickly by comparing vector to vector, which is something the logic layer can do very efficiently using some logic gates, right? And that's it. For Grimm filter, this is the main idea, and this is how the algorithm works. So you get the read, you extract all these sequences, and you map them to a corresponding bits. For the reference genome, this can be a pre-processing step. You do it once for the reference genome, because the reference genome doesn't change unless you change the species. You move to viruses, for example. But for the read, it is included in the runtime. It's not a pre-processing, right? So it's part of the runtime. Once you generate this bit vector, you count how many ones, and then you compare it to the ones you have from the reference genome. You could do it in another way. Instead of adding up all the ones and then count how many matches you get, you could just compare one by one. So you do comparator between the vectors and see how many times you have a matches. And then you count the matches and you see if it is above a threshold or not. That is one of the simplest way you can do for the filtering. However, as you can see, the simplest you go, the higher false positive you will generate. So there is always trade-off. You cannot go with very simple algorithm solving all your problems, right? But it was optimal for the hardware, for the architecture we have, where we can lay out, if we do the layout optimally as to fit many data in the 3D stacked memory, this is where we get the most benefits out of this hardware. That's why it's important to design an algorithm that is aware about the hardware that you are using. All right, I think, this is how it works, how you map these to the reference genome. And then once you get a hit or enough number of matches, then you calculate the edit distance at the CPU side. As I said, we never replace the edit distance, because edit distance is the most optimal algorithm used to find the differences. So you cannot replace it by anything else. But what you can do is reduce the number of times that you can use it. That is a Grimm filter paper, if you're interested in it. There is also Genome. In Genome, we do near memory acceleration for sequence alignment. Let's escape this. One important thing to mention, there is an algorithm called BiTAP. This is a very old algorithm proposed in the 90s, about 30 years ago, okay? In that time, it has some limitation. Nobody used it after that. So it was proposed and then nobody used it because it has limitation that cannot be affordable. We solve these limitations and we make it supporting long reads, support traceback, which is very important to tell you where are these differences to provide the cigar string, and also make it parallelizable using near memory acceleration. And the results were real significant. And since we developed a sequence aligner algorithm, we can still use it as a pre-alignment filtering, as in Sneaky Snake, for example, because we can do filtering, but very optimally, because we can provide the exact number of edits needed between the two sequences. Yes, please. Excellent. Excellent observation. Yes, ultimately. For example, these devices, these nanopore sequencing, right? The very expensive piece, right? So this is nanopore technology, produce electrical current, right? And what we use for that machine learning algorithm. And this machine learning algorithm is used in text search. And we use it here for base calling, just to convert the electrical signal into text. That is QuartzNet architecture, if you know in machine learning. There is also LSTM architecture for doing the base calling. So there is a lot of overlap between multiple fields. And that's the best thing if you are aware about similar problem, then you can leverage this algorithm to solve this problem in bioinformatics, for example. Yeah. So computing is still bottlenecked by data movement for all these accelerators, because whatever the accelerator we use it, even if it is near cache or near memory, you still need to move the data from the storage to these devices, right? To the memory device, or from the sequencing to these devices. And this is our latest work, and hopefully this is the last work to present, called GenStore. We presented this month in Lausanne for S+. And what we try to do here to accelerate read mapping inside the storage or near the data itself, so inside the SSD. And there are two main observations here. The first one is that we find enough number of exact matches between the read and the reference genome. So instead of checking the edit distance calculation and all those sequence alignment expensive algorithm, we find large portion of the read exactly appear in the reference genome. And this is logical for short reads, because short reads, the accuracy is very high from the sequencing machine, and individuals from the same species or same organism, they are always similar to each other, right? There is 99.9% similarity between them. So if you get reads from these regions that are similar, then you expect most of the reads to exactly match over there. And we leverage this observation for only short reads, not for long reads, to do the filtering quickly inside the storage. So in the storage, you can fetch a huge page of data, and then you can do the process in bulk, if it is allowed. That's why it's very important to design algorithm for this architecture or the SSD. The second observation is that for long reads, now we want to address the problem. What we observe that they share always large number of short, exactly matching subsequences. So for short reads, they share the entire read as is. But for long reads, they share large number of exact matches subsequences, very short subsequences. For example, 16 long subsequences. If you find this number of exactly matching subsequences above a threshold, then it means probably these two sequences are similar. If not, filter them inside the storage. There is no need to move the data from the storage to main memory, to the caches, to the CPU. And that's it for GenStore. The speed up is significant end-to-end. You are still doing read mapping in traditional way for the other reads. For example, if the short reads are still not matching exactly, still there might be interesting, right? There might be single difference or two differences. That's still significant. So you need to move it to the CPU and do read mapping only for those few reads, which is very few percentage of the total number of reads. If you're interested in this, there's a large video, I think about 20 minutes on YouTube. We published yesterday. So I would recommend everyone to check the link down. The slides, by the way, already as usual on the course webpage. So you can check the video. Now I'll tell you just the last story about adoption of these hardware accelerators in real life. Now you have all these genomics companies around the world and hospitals everywhere. How about using these hardware accelerators, either proposed by us or proposed by other research groups? We published one of our earlier paper in December 2016, and we got this review from one of the reviewers. He said, there are a lot of hardware accelerators, but there is little to no adoption of previous specialized hardware solutions related to improving the speed of the alignment. What he's saying, okay, this is not a problem with the work itself, but a problem with adoption. You are proposing a lot of hardware accelerators, but then nobody is using them. What is the goal? And this is what we answer. We tell him or her, it always takes time to adopt new or different hardware technology since it requires investment into the hardware infrastructure. And we were the first to propose that work using FPGA. Nobody used FPGA for filtering before. So we tell him, people in the community will take time to digest the need for FPGA because most of them might prefer to have C programming, a code that they can change later on, play with it, improve it. But if they have an FPGA, they need to understand the logic and then recompile, synthesize it, and move the code to the FPGA. If it is CMOS, for example, they cannot do anything. They just need to trash the chip, design a new one, right? So that is something challenging, actually, to address. We have the long response attached in the slide, so you can look at it later. We were at that time dreamer in 2016. Nobody used these hardware accelerators for genomics in real life. But after that, in 2018, we start to see some of these accelerators happening inside the sequencing machine itself. So coming from the companies produce these. This is one of them called Dragon. That is for Illumina devices. There's another one from NVIDIA that is already used in the nanopore for base calling. So they have 4 GPU cards inside one of the machines, Promethean. And you can see the speed up is really huge. Moving forward, what are the possible options to see in the future in this sequencing machine? We have a specialized chip to do machine learning, AI, right? We have also other chips to do self-driving. How about genomics? Why we don't have specialized devices to do that? Luckily, yesterday or just a few days ago, we got a chip from NVIDIA. This is the latest GPU card we have called H100. What they have in this card, they have a specialized core just to perform dynamic programming, to do one of the most time-consuming steps in genomics, which is sequence alignment. And you can see 7x improvement just because of the specialization, just by having that. So you can think about the future of genomics by having all these big companies, big heads to move towards specialization as we have in machine learning, AI, and so on. There are a lot of things going on in the news. You can read it later on, but you can see most of the companies start to acquire hardware acceleration startups. So if you are smart people, want to build startup to accelerate things in GPU FPGA, this company will look for you. This is one of the first sequencing machines that they have analysis capability inside it. So it's no longer only sequencing, but also the analysis happening inside the machine. This is Illumina NextSeq 2000. This year, we got a huge grant from European Union with the consortium with collaboration from our group, including our research group, just to accelerate genomics near memory. So we are going to profile a lot of applications to see their suitability for being analyzed or processed either inside the DRAM or near the memory. And we are going to use one of the technology. We have this French startup in our consortium. They're helping us with this to do the analysis inside real hardware. So this is a real DRAM module. You can see these modules fitted inside as if it is traditional DDR. This is also from ETH. They are trying to use the DNA as a storage material. So this is synthetic DNA. This bunny shape is synthetic DNA. It's manufactured. And within the DNA, if there's only 4 possible options, then you can encode any data using 2 bits, right? The A, you can consider it as 0, 0, the C as 0, 1, G as 1, 0, T as 1, 1, right? By knowing this information, then once you read the DNA of this bunny or this synthetic material, whenever you have A, you convert it to 0, 0, C into this. Once you get this full content of binary data, you can convert it as whatever encoding you were using to fetch back the data you were storing. However, there's a challenge here. The sequencing error still cannot read this content accurately. Then you need to do redundancy, right? You redundant a lot of information. Once you read it using the sequencing machine, you can overlap them and get a correction for this information. That's why we cannot store too many things inside this bunny. So they use about 45 kilobyte of data, here 1.4 megabyte inside the lenses of the sunglass or the glasses. So they make synthetic DNA out of this material, okay? So the information are already embedded here. You need to do, of course, library preparation to fetch the data, then sequencing, and then some correction, some read mapping, and then you get the data. But the very good thing about this, how durable this information. You cannot think about the endurance or the lifetime of this material compared to memory, for example, or storage. This can stay for ages, untouched. If you don't melt it down, of course, if you don't play with it, this can stay for thousands of years, as is, compared to SSD or other type of memories. Yes, please. That's very ambitious. I hope so. I hope so, to have this in reality. You can do personalized shopping. For example, you have your DNA read already. You know what kind of genes you have, and then you know what kind of product are suitable for your DNA. Instead of buying anything randomly, and you may have allergy to these kind of things, you just do scanning by your smartwatch to some barcode, and then the smartwatch will tell you if this is suitable for your DNA or not, or for your health. This is a real company. You can do microbiome profiling and so on. And now most of the countries start to do sequencing for their own population. Now, this is the question we answer. Some of you ask me, what if we have a machine that gives you the full sequence as DNA? I think I answered this question. You can do it for comparing genomics, for genomics, metagenomics, and pangenomics. And to conclude the talk, we still have this very challenging question, how and where to enable fast, cheap, privacy-preserving exabyte scale of genome analysis. We still don't know what is the best way to do it and where to do it, whether inside the sequencing, inside the storage, main memory, and so on. This is very important. I mentioned it multiple times during the talk. The speed up should always come from new algorithm and new hardware. That is the best way to do it. And next, this just references pointer on reading materials. This is feedback we received from the community in one of our paper. I recommend everyone to read this first before the other papers. This will give you a full overview of everything on read mapping. There are a lot of videos available online. We are offering 3 courses, 2 of them on genomics, very specific, and the 3rd one on heterogeneous system to accelerate bioinformatics application. If you're interested, you can join the course or you can watch the video. Contact us if you want a project to assign to you so you can get more involved in this area. Thank you so much. We exceed the time by 4 minutes. Sorry for that. Yeah, if you have any question, I'm happy to take them now. If there are people on Zoom, sorry, I didn't notice if we got any messages on Zoom or YouTube. I think Joel can tell me if we got any question on Zoom. No? YouTube? Okay, perfect. I can take still questions. Otherwise, see you next week. Thank you so much.